Let's begin to discuss now genetically what's going on when you have an organism that is 2n plus 1, so a diploid set plus one extra chromosome, that attempts to go through meiosis. I've drawn this now with a chromosome that has just one locus so that we can ignore the effects of crossing over. Crossing over is very difficult to handle in a situation where you have synapsis now, or attempted synapsis between three homologous partners within one cell. So the first stage will be chromosome replication as before. Now when it comes to synapsis, there's a lot of different possibilities. You could have any two chromosomes find each other in synapse, leaving the third one completely unsynapsed. That's one option. Other possibilities exist, and there are sort of extremes on either end and then different levels of synapsis. But one option is that you can actually have partial synapsis, say between these two, and then synapsis just based on what sections of the chromosomes find each other in synapse first. You could have this sort of setup where you have three chromosomes partially synapsed with one another. The issue is that the cell for meiosis 1 is simply going to be looking in and saying, I want one of each centromere for the synapse pairs that are at the middle of the cell, and I'll take one of each of them, but this third one that is not synapsed will have a 50-50 shot going one way or the other. And the same sort of thing happens here. Here's the synapse centromeres. The cell says, I want one of each of those. And then this one has a 50-50 shot of going one way or the other. This all gets very complicated, or it seems like it can be very complicated. But there's a very handy way to summarize what's going on in a 2n plus 1 meiosis. The thing to realize is that for every n gamete that's formed out of this, there will be an n plus 1 gamete that is also formed at the same time. So for example, if I have a gamete that is formed that just has this chromosome with a little d on it, the n plus 1 gamete will have these two chromosomes. If I take simply one of the big D chromosomes alone, then that's what's on the n plus 1 side, and there are two ways to do that. So these are the possible outputs, and simply because this is all based on independent assortment and 50-50 probabilities, each of these will be equal in probability and have a frequency of 1/6. And in the next segment, what we'll do is we'll test cross this organism, this 2n plus 1 organism, which is forming products of meiosis, say gametes, and we'll take this and we'll test cross it to a normal organism that is little d as a 2n. And we'll do that in the next segment. Okay, we were examining this case of an organism that is 2n plus 1 with chromosomes like this, making gametes. So this is undergoing meiosis as best it can to produce these different outputs. Now these are products of meiosis that are either n or n plus 1, as we've been discussing. Now this normal 2n organism is going to produce products of meiosis or gametes that are simply little d and n. So now what would we observe when we do this cross? If this in fact was a 2n plus 1 organism, what we would notice is that 5/6 of the progeny have at least one big D allele. So working on the assumption that one big D is enough to give you whatever phenotype that is, you would see this sort of thing. Only 1/6 would have the little d phenotype now. And the reason for that is only 1/6 of the gametes coming out of this 2n plus 1 organism have the little d alone. Everything else has the little d with a big D along for the ride. The other thing we would see, if we have some way of detecting it, is we would see that 3/6 of the total products of meiosis are 2n plus 1. Sorry, 3/6 of the total progeny resulting from the fusion of the different products of meiosis from this organism and this organism. 3/6 are 2n plus 1, and 3/6 are simply 2n. So it's a 50-50 mix between 2n plus 1 progeny, taking this little d and fusing it with these, or simply taking the n gametes and fusing them with this. And you could do a full breakdown of the different genotypes. You would have 1/6 that are little d, 1/6 that are big D, little d, another 1/6 that are big D, little d, so those would go together as 2/6. Then you'd have another 1/6 that are like this. Okay, so there you go. That would be the full output. And you would see some progeny, half of the progeny would be 2n, half the progeny would be 2n plus 1. So where you would see a difference relative to a cross where, say, this was just, let's say perhaps you expected this to be simply a 2n organism that was big D, little d. Say there was a non-disjunction event that put two big D's together. You were expecting this organism to be 2n. Unbeknownst to you, it's 2n plus 1. If you took this and you crossed it with little d, you would simply expect 1/2 big D, little d, and 1/2 little d. Just simply that 50-50 ratio, that monohybrid ratio that you're used to. Now we have quite a different ratio. We have a 5/6 to 1/6 ratio phenotypically, whereas here we had a 50-50, say wild type to little d phenotype. Now we have 5/6 that are wild type, at least genetically for the D locus, and we have only 1/6 that show the recessive phenotype. So this 5/6 to 1/6 ratio is a tip-off that you may well be dealing with a 2n plus 1 organism. You're probably familiar with at least one 2n plus 1 case, and it's a case in humans that you're most likely familiar with. The technical name for a 2n plus 1 situation is something called trisomy, or an individual who has a 2n plus 1 state is said to be trisomic. You're probably familiar with trisomy 21 in humans. This is 3 copies, or 2n plus 1 for chromosome number 21 in humans, and this is also known as Down syndrome. Now, all the different possible trisomies in humans do form. So there's trisomy 1, trisomy 2, all the way up to trisomy 23, and also trisomies that can affect the X and the Y chromosomes as well. The reason why we only observe trisomy 21 and a few other ones, say as trisomy 18 is another one and trisomy 13, is that very few of these trisomies actually survive past birth. Many of them end as miscarriages. Trisomy 21 is one of the ones that is the most viable, and Down syndrome is the resulting condition that one gets. Now, the reason why there's a condition, why there's actually a syndrome, has to do with gene balance. We've been talking about the importance of having no duplications or deletions in products of meiosis, and for the most part, in most organisms, extra copies of genetic material or a deletion of genetic material ends with non-functional products of meiosis. But that's not strictly true. Sometimes extra copies of a different chromosome, or sometimes even loss of a chromosome, can result in a state that is viable, and it is something that is an aneuploid. Aneuploid simply means something that's less than a perfect multiple of the haploid number. So something that is not a perfect multiple of n. So in the case of a trisomy, it's not a perfect multiple of n because it's 2n plus 1. There are some other chromosome aneuploidies that we'll discuss in the next segment. Here are some of the human aneuploidies that are known clinically, although as I mentioned before, genetically speaking, all of the human aneuploidies do form at some frequency. It's just a question of how long they survive in utero or post-term. Trisomy 13 and trisomy 18 are ones that do survive post-term at a small frequency. Trisomy 21, as we mentioned before, is Downs. Turner syndrome is a aneuploidy that is actually an X chromosome with nothing paired with it, so an X and then lacking a second X or a Y. So this is an aneuploidy that is a 2n minus 1 state. These individuals are phenotypically female but are sterile. Klinefelter syndrome, on the other hand, is another sex chromosome aneuploidy in humans. This is the presence of two X chromosomes plus a Y chromosome. So this is another 2n plus 1 state. Individuals with Klinefelter syndrome are phenotypically male and in many cases do not have symptoms that would be clinically recognized, although it's a range. Some individuals do have some fertility problems and others do not, so it's quite a range. This is actually the most common sex chromosome aneuploidy in humans, and many individuals who have this condition would not even necessarily know. So there's a range of different possible conditions in humans, and we'll examine some of these in questions in class.